morning, students. In this particular lecture we are talking about the types of the hearts if we take them on the basis of the morphological aspects. So here students, if we talk about the types of the heart on the basis of the morphology, so we have got a major 4 types of hearts, and these types, they are, if we talk about the types on the basis of morphology, the first would be, these are the chambered hearts. After chambered hearts, we have the second category of the hearts, and these are the tubular hearts. Now that can be considered as a second type of the heart. Now these are long tube-like hearts which may be present in, these are relatively present in the invertebrates. Now if we talk about the third type of the hearts on the basis of the morphology, these are the pulsatile, you can say pulsatile vessels. So these are the pulsating vessels which are present in some of the animals, and those can be considered as the type of the heart in those animals. Now if I go further, we have the fourth type of the heart, and these are the ampullary hearts. So students, these are the 4 basic categories of the types, you can say, of the hearts on the basis of the morphology. We have also taken the type of the heart on the basis of the you can say the origin of the heartbeat, and those will be the neurogenic and the myogenic hearts. So those will also be taken in that particular lecture, but here we are considering the types of the heart on the basis of the morphology. So students, we'll be taking these types of the hearts one by one. So I am starting with the very first type, which are the chambered hearts. So students, if we talk about these chambered hearts, these may be found among the vertebrates, and they are also present in some of the mollusks. So we'll be taking these hearts one by one. So first of all, we are starting with the chambered hearts of the vertebrates. Now here, the vertebrate hearts, they have got different number of chambers, and they are equipped with valves. As we have very you can say in a wide description, we have taken the types of the hearts in the particular question of the evolution of heart. So there we discussed that how the chambers, they have increased in number, and what is the you can say the status of the interventricular septum, and many of the hearts, they have got partial septum. So we had considered them as 2.5, or we are considering it as 3.5 chambered heart. So the sequence of the contraction is essentially similar in both the two-sided, which can be the 3 or 4 chambered, and one-sided hearts. So these are the 2-chambered hearts. That means the vertebrates, they may have one-sided kind of the hearts, which are having only 2 chambers. One is the auricle, one is the ventricle, and there can be 2-sided hearts, which are the 3 and the 4 chambered hearts, which have got double circulation kind of mechanism. Now if we start with the fishes, so in the vertebrates, now we are starting with the fishes. So if you take the case of the fishes, what we have is there is a single type of circulation, and we have got a single auricle. So here we have one auricle, and one ventricle. So we have got two chambers in this particular you can say group, and the blood, it passes through the gill vessels before entering into the aorta, into the dorsal aorta specifically saying. So what will happen, that it will be having two chambers. So one will be the you can say the auricle, then we'll be having a ventricle, and here the ventricle is smaller as compared to the auricle. Now after the ventricle, we have got some accessory chambers also present in this group in the fishes. So we have got here the I'm using blue color for this. So this will be the conus arteriosus, and here we have got a sinus venosus. So these are the two accessory chambers. So here we have this is the auricle, this is the ventricle, this will be the sinus venosus, that is an accessory chamber, and here we have a conus arteriosus. So these are the major two chambers, one is auricle, one is ventricle. Now the oxygen depleted blood, which is deoxygenated blood, that is returning from the body, it enters the atrium, and then the blood, it flows to the ventricle. So it is pumped from the ventricle and it goes to the ventral aorta through the conus arteriosus. So whenever it is going from the body, first of all it will reach the sinus venosus. From sinus venosus it will reach the auricle, then to the ventricle, then through the conus it will enter into the ventral aorta. So on the top here we can write as, this is the ventral aorta in which we have the deoxygenated blood. So students, from the ventral aorta, this deoxygenated blood is going into the gills. So if we go further, it will lead to the gills. So here I'm writing as the gills. So whenever it is reaching the gills, it will be oxygenated. So this oxygenated blood, it will go to the body organs, and then it will come back to the auricle and ventricle. So what I want to say over here is that you are not going to give the circulation mechanism or how the circulation happens, but we are concerned with the chambers of the heart over here. So the heart of the cartilaginous dogfish is muscular and dorsoventrally bent in the S-shaped tube with 4 compartments in a linear series. So as you can see in the diagram also, so this is a linear series of 4 chambers. 2 are the main chambers and 2 are the accessory chambers. So we are not talking much about the type of the heart over here. So if we go further, we have the case of amphibians now. So in the amphibians, we have got a 3-chambered heart. So what we have over here is that we have you have 2 auricles and 1 ventricle. So that means the auricle has been divided into 2 chambers, and we have an intra-auricular septum present over here. So what happens over here, that the heart, it is taking up the oxygenated and the deoxygenated blood into the heart. It is taking both the types of the streams, but they are not separated in the ventricle because we have got a single ventricle. So the blood, it returns twice to the heart for each of the cycle in this particular case. So in the frog, if you take a frog as a representative animal in amphibians, so the isometric systole, it can last for about like 0.13 seconds. And apparently some blood, it remains in the ventricle after each ejection. So this is a very small period. So the dorsal atrium, it shifts anterior to the ventricle. And the sinus venosus here, it opens into the right atrium dorsally and not posteriorly. So that means we have got the sinus venosus in the connection with the right atrium only. So there is no connection with the left atrium of the sinus venosus. So the atrium, it is completely divided into right and left chambers and has no kind of foramen ovale in the interauricular septum. So they are fully divided. So we are talking about these two auricles. So they are fully divided. The septum is complete. So if you take the case of the dipnonians, so here we can have the foramen of ovale in this particular group, where uh, the septum in between the two auricles, they remain open, uh, you can say for some magnitude. So in that case it is not completely divided, so we can say like this. So deep pockets, they develop into the ventricular cavity. So the accessory chamber, which is the conus arteriosus over here also, so it divides into the systemic and the pulmonary vessels by a spiral valve. So here we have a spiral valve also. But if we talk about the chambers of amphibians, we have got two auricles and we have got a single ventricle. So that is again a chambered kind of a heart. Now students, if I go further of the amphibians, now we have got the reptiles. And if we talk about reptiles, what we have is the establishment of a low pressure respiratory blood shunt and the consequent high systemic pressure aided the reptiles to withstand the rigors of the terrestrial life. As reptiles are fully terrestrial animals and they are living on land, and the forms of the reptiles, they are like varied. They can be of very small sizes and they can attain a very long, you can say, length, as we have seen in the crocodiles. So what is the condition in reptiles? That we have the atrium as completely divided into two. So here also we have got two atria. So we've got two atria. And what about the ventricle now? So it is partially divided. So we have got one ventricle, but it has got the partial division. So in the case of reptiles, we can have a 3.5 chambered heart, typically speaking. The ventricle is partially divided by the septum, but there will be no mixing of blood of the two streams in this particular case because of the presence of the spiral valve. So that is a good thing over here. But in reptiles, this mixing is, you can say, prevented by the formation of some rugae-like structures in the ventricle. So in the amphibians, we had the spiral valve, but in reptiles, the function is performed by these rugae-like structures in the ventricle. So the ventricle is also partially divided by a septum in most of the reptiles, but in the case of crocodiles and the alligators, we have got a four-chambered heart. So this is an exception kind of a thing in reptiles, that in the crocodiles and the alligators we have got a four-chambered heart. So this is very comparable heart to a bird or a mammal. So that means that oxygenated blood coming from the lungs, it will go to the left side of the heart as we have, and it is essentially separated from the deoxygenated blood which is going to the right side of the heart. So if we talk about the reptiles, we have got a full, you can say, a channeled blood system. So here we have got two streams of the blood and they are kept separate in whole of the heart in the reptiles. Now after reptiles, if we talk about the next part, or the next group I should say. So these are the birds and the mammals. Now here in this particular case we have got two auricles and two ventricles, as we have got a complete septa in both the cases. So they are complete hearts. As per the evolution, they are the modified hearts, and they are capable to keep the two streams separate to an efficiency of 100%. So in the case of mammals, the entire sequence in the vertebrate heart consists of the successive contractions in the first auricles, then they are going to the ventricles. The ventricle here is completely divided into two. So the heart is four-chambered, having the two auricles and the two ventricles. So there is a complete separation of the venous and the arterial blood, as we have discussed earlier. The systemic aorta, it is leaving the left ventricle and it is carrying the blood to the head and the body. So it is going for the head through the carotids and the body through the systemics, while the pulmonary artery, it is leaving the right ventricle and it is carrying the blood to the lungs for oxygenation. So that is the kind of aortic arch, you can say, plan in the case of birds and mammals. There is a double kind of circulation over here in birds and mammals. So this is a typical double circulation, and there is no mixing of blood at any place in these hearts. The sinus venosus is completely incorporated into the right auricle over here. So as we don't have any kind of sinus venosus in ourselves, in mammals or in birds also, we don't have sinus venosus because it is incorporated into the right auricle, and now it is represented by the valves which are present in the right auricle. So the you can say the atrial valves, they are representing the sinus venosus. Now the right auricle here, it is receiving the two precables and a postcable also. So the left auricle, it receives the oxygenated blood through the pulmonary veins. The conus arteriosus is absent over here, as the left ventricle is quite muscular as compared to the right ventricle, and it has got the capability to pump the blood to whole of the body of the birds and the mammals. So a single systemic aorta, it arises from the left ventricle, and both have the valves at their bases. So we have got bicuspid and tricuspid valves also in the case of mammals and birds, but the systemics are on the opposite sides in the case of birds and mammals. So that will be taken in the particular topic of the evolution of the heart. So here, students, we have a overall idea that if you have got a question on the chambered hearts, then you have to take these animals first of all into this particular question. So students, if you are getting this particular question in your examination, you can give this kind of structure in the chambered hearts. So here you can very well see that we have a heart in the case of the fishes. We have got a two-chambered heart. In the case of amphibians, we have got a typical three-chambered heart. So here you can very well see that the auricles, they are totally separated out, and here we have a ventricle. So this is a typical three-chambered heart in amphibians. And if you take the case of the reptiles, so here also we have got two auricles and a partially divided, you can say, the ventricle. So typically it is a 3.5 chambered heart in the case of reptiles, and if we go for the mammals, you have got a typical 4-chambered heart which is having two auricles and two ventricles. So you have to make these kinds of diagrams if you get a question on the chambered heart. So you are not going to give the full internal structure of the hearts in every group. So students, after this particular part, we are taking up the second type or you can say second group of the chambered hearts. In the very first group, we had taken the vertebrates. So students, after the vertebrates, we have got the chambered hearts in the mollusks also. So the chambered hearts of the mollusks, it consists of either one or two auricles, and if you have got two auricles, there can be one ventricle. So there can be one auricle and one ventricle, or there can be two auricles and one ventricle. So the auricles, they are usually receiving chambers with the little of the musculature, as we have seen in the case of fishes also, whereas the ventricle is a strong contractile part of the heart which is making the blood pressure. The systemic heart of the cephalopod mollusks is particularly well developed, and the blood, it returns from the body by veins to the branchial hearts at the base of the gills and passes from the gills in veins to the auricles of the systemic heart. Now this arrangement, you can say, these are also called as the booster hearts in the case of mollusks. So these hearts, they are proximal to the gills, and it is more efficient than in the plan of the fishes, in which the heart has to overcome the resistance of the vessels in both the gills and the body tissues. So the muscles of the molluscan hearts, they are particularly, if we speak about the cephalopods and gastropods, they are striated. So that means they have got a similar kind of muscular platform in their hearts also. So students, in mollusks, first I am taking the example of the unio. So unio is a bivalve, and the heart of the unio, it consists of a muscular ventricle and two thin-walled auricles. So here we have two auricles and one ventricle in this particular case. Now this whole of the heart, it is enclosed by a pericardium. So this is the layer which is you can say wrapping the whole of the heart. So the auricle, it communicates with the ventricle, and this opening is guarded by the valves in this particular case also. So the two aortae, they originate from the two ends of the ventricle. So this can be seen in this particular diagram. So here students, we can see the heart of the unio. So here you can very well see that this is the ventricle, this is the ventricle, and this is the auricle. So what we have over here is that the two aortae, they are originating from the two ends of the single ventricle. So here you can very well see the aortae, they are originating on both the ends of the ventricle. One is the anterior aorta, which is situated above the rectum, and the other is the posterior aorta, located below the rectum. So students, here you can very well see that this is the stomach part. So this is the anterior aorta, and this will be the posterior aorta on the right side here in this particular diagram. So overall, if we talk about the unio, here we have got two thin-walled auricles and a single ventricle. We got two aortae, we have got the pericardium, as we have in humans also. So that was the case of the unio. Now students, after unio, we have got the second example of pila. Now here in this animal, we have got a single auricle and a single ventricle. So these are the two chambers which are present in the pila. So the heart, it consists of two chambers. One is the auricle and one is the ventricle in this case. The auricle is thin-walled, and it is highly contractile, and it is roughly triangular in shape, and it is situated in the dorsal part of the pericardium. So here you can very well see this is the auricle in the case of the pila, and the dorsal part of the auricle, it receives the blood from three main veins. So as you can very well see, so these are the three main veins through which the auricle is receiving the blood. And ventrally, the auricle, it communicates with the ventricle from here, and it is through the auriculoventricular aperture. So this is the aperture by which the auricle is opening into the ventricle, and the blood flows from the auricle to ventricle. Now, the auriculoventricular aperture is guarded by the semilunar valves, which prevent the regurgitation of the blood or the backflow of the blood from the ventricle to the auricle. So this is the main function of these valves. The ventricle, if we talk about the ventricle of pila, it is ovoidal sac-like structure, and it is lying below the level of the auricle. Its wall is thick, somewhat spongy, and it is muscular also. So the lower end of the ventricle gives rise to a large artery which is called as the aortic trunk. So this is the basic cephalic aorta which is coming out of the heart, or the ventricle, I should say, over here. So it is making, this is the ventral aorta which is going to the viscera, and this is the cephalic aorta which is going to the head part, and here we have the gastric artery, and this is the main visceral artery which is coming out of the ventricle of the pila. So that was about the heart of the pila. So students, after this, if we talk about the overall idea of the chambered hearts as the type, as the morphological type of the heart, so we have to describe first of all the types of the hearts in the case of the vertebrates, and secondly we have to take the case of the mollusks. So if you comprise all these types into a single question, that can comprise the chambered heart question in your examination. So I hope you have gone through all the aspects of the chambered heart, and you are making very simple diagrams in this particular question.